Hello, and welcome to Human Anatomy and Physiology 2. Today we will be starting our 8th unit, Metabolism and Nutrition. The objectives of this unit include understanding the absorption of carbohydrates, lipids, and proteins, identifying similarities and differences in the metabolic pathways for carbohydrates, lipids, and proteins, and assessing general dietary habits using the USDA's SuperTracker online tool. We talked about digestion in our previous unit. As we discussed previously, the goal of digestion is to break down food physically and chemically. Its physical and chemical breakdown allows the small intestine to absorb key nutrients, carbohydrates, lipids, and proteins. Once absorbed as monomers, reassembly occurs to form polymers for storage, which can then be used at a later time. It is the breakdown of these polymers that will generate energy, or ATP, for utilization. In this unit, you will review chemical digestion, including storage and usage, as well as metabolic physiology. As we learned in our previous unit, digestion begins with mechanical digestion, which is the physical breakdown of food from chewing. In the food being chewed, there are many different nutrients, complex carbohydrates, proteins, and lipids. These are called macromolecules and are further broken down by chemical digestion. It is this type of digestion that allows the macromolecules to be made into fundamental building blocks that can be used by the body's cells. In order for these macromolecules to be further broken down, enzymes have to be present. There are a multitude of enzymes that perform specific functions in breaking down macromolecules into their very specific component parts. The figure seen within the Overview of Chemical Digestion section is helpful in knowing what these enzymes are, as well as their functions. Certain biological molecules, or macromolecules, obtained from the food consumed is actually stored based on your body's needs. Glucose, for example, is stored as glycogen within the liver and skeletal muscle cells. As for fatty acids and glycerol, they are combined and stored as triglycerides in the liver and skeletal muscles, and even adipocytes. So, what about amino acids? There actually is no storage form for these biological molecules, but they are used as building blocks within cells to manufacture proteins. Do note, though, that they cannot be converted into energy until they are deaminated, essentially until an amino group has been removed. A flow diagram of metabolic physiology can be seen in your laboratory manual, following the section regarding storage. Use this to further your understanding of how macromolecules are stored. Alright, so we've talked about how they're stored, and we know that the ultimate goal is for our body to use them as macromolecules. Simply put, regardless of the pathway, the ultimate goal of these steps is the creation of acetyl-CoA. This can enter the citric acid cycle and eventually the electron transport chain. By the end of these processes, carbon dioxide and water are created as waste products. We've talked about chemical digestion for a bit now. Let's talk about metabolic physiology. For carbohydrates, there are two phases to glucose breakdown. They are anaerobic and aerobic glycolysis. The terms simply identify the phases of catabolism in the absence, anaerobic, or presence, aerobic, of oxygen. With anaerobic glycolysis, or fermentation, pyruvate remains anaerobic and produces lactate. It is this lactate that can be converted back into the pyruvate and even further converted into glucose. For the breakdown, lipolysis, or creation, lipogenesis, of lipids, do note the figure provided. This figure shows these proteins in the form of a flowchart, making it particularly easier to understand. Amino acids, on the other hand, can be stripped of their amino group, as we mentioned previously. and processed for energy or converted from one amino acid into another. This is done through transamination, seen in the figure provided. It is important to note that any free nitrogen-containing compounds have to be processed through the urea cycle and excreted in urine from the kidneys. At the end of this unit, you will notice review questions covering the material learned. Chemical digestion, including storage and usage, as well as metabolic physiology. Be sure to fill these out thoroughly using the knowledge you've learned, as these processes could surely be seen on your upcoming practical. Thank you for joining me as we started to investigate metabolism and nutrition. Have fun as you continue to adventure further, and I'll see you for the next unit.